Howdy, this is Browdy, coming at you with the fourth and final video lecture for the evolution/history of life combined unit. So we've started first and foremost by talking about evolution and natural selection and what those are. Second, we looked at different kinds of evidence of evolution. Third, most recently, we talked about different evolutionary patterns and cladograms and cladistics and just the classification of living things. And so lastly, we're actually going to talk about the history of life on Earth and really a number of things in terms of how we believe we got from where we started to where we are today. So let's dive right in. So first and foremost, now that's not a very good looking timeline. Before I talk about the nebula hypothesis, actually, I'll point out here in the notes, we have like a very quick timeline of Earth. And I mentioned this in class, so I'm going to mention this here. Now, these are approximate dates. One of the most important is that I want you to know, obviously, that we believe the Earth itself was formed about 4.6 billion years ago. Well, that's a really long time. Think about how long the average human lifespan is. Typically, it caps out around about 100, right? And 10 of those would be 1,000 years. 1,000 times 1,000 is 1,000,000. 1,000 times 1,000,000 is 1,000,000,000. And so we think 4.6 billion years ago, the Earth was formed. And the way that we believe that happened was what we call the nebula hypothesis. Now, I won't pretend to be an astronomy or geology expert with a lot of the stuff we're talking about today. So these are going to be some pretty kind of brief surface level explanations, but I think it's enough to kind of get the point across for now. And so the way we believe the Earth was formed is actually what we refer to as the nebula hypothesis. And so as you may or may not know, that a star out in space has what we would consider to be a lifespan. Now, that's not to say that it's really alive, so to speak, but it has a lifespan from the point where it's formed to the point that it explodes. And at the end of a star's life, it explodes. And so we believe that an exploding star created this rotating circle of dust. So there was some movement to it, dust and debris. And we know that things that have greater amounts of mass or greater mass have a greater gravitational pull. And what we believe is that most of that mass was pulled to the center by gravity and formed what we now have as the sun. And the remaining rocks and debris and particles and things like that condensed again through gravity into what we now have as the planets. And so that we believe is the origin of our solar system. Again, it's a very hard thing for us to test, but currently that's the leading hypothesis, and we call that the nebula hypothesis. And so we think that 4.6 billion years ago, the Earth as we know it came together. Now, going back to that timeline, that was 4.6 billion years ago. And again, these are approximate ages for the sake of the notes, but it wasn't for another 1.1 or so billion years that we believe life itself first formed, 3.5 billion years, giving us prokaryotic cells. Remember that prokaryotes are less complex than eukaryotes, right? Prokaryotes have DNA, a cell membrane, cytoplasm, and ribosomes, but not nucleus, not organelles, not really any of the complex stuff. They're single cells. So we think prokaryotic life started about 3.5 billion years ago. It wasn't until 2.1 billion years ago, so again, a huge jump, that we think eukaryotic cells evolved. It wasn't until about 700 million years ago that we saw multicellular life. Then 540 million years ago, we had what we call the Cambrian explosion of life forms, where life just suddenly diversified greatly. So we had seen very minimal evolution up to that point, but once we had multicellular life, we really had what we needed for things to kind of take form and run with it. And so the Cambrian explosion is what we refer to as really one of just the biggest mass evolutions of different kinds of life. 420 million years ago, we believe we had land plants evolve. 360 million years ago, land animals, which we believe were amphibians. 230 million years ago, the dinosaurs. 140 million years ago, flowering plants. 65 million years ago, we believe was the last, most recent mass extinction, which is the one that killed the dinosaurs. And then it wasn't until very, very, very recently, in this grand scheme of things, that humans emerged, and even more recently that humans have had recorded history. So one thing that's important to keep in mind, especially if you've seen the videos in class that kind of put Earth's timeline into perspective, we've been around very, very, very little time in the grand scheme of the Earth. Yet, we figured out a whole lot, or at least we believe that we have. And so, while that's a really intimidating time frame, it's also encouraging to think about how far we've come in so little time. So, take that for what you will. Let's keep going. Now, there's some key requirements for life to exist. So if that's when Earth started, 4.6 billion years ago, we think through the nebula hypothesis, or that's our leading theory of how the Earth might have been formed. Let's talk about how life might have been formed. So we think that there's really two key requirements for life. First would be organic molecule building blocks, so things like nucleotides and amino acids. And second, some way to keep the insides of living things separate from the outside environment. So some sort of divider or membrane, something of the sort. So first, let's talk about the two most popular hypotheses for how the molecules of life were formed, so how organic molecules came to be. The first is the Miller-Urey experiment. Now, the Miller-Urey experiment is famous because it was one of the first examples of synthesizing, creating organic molecules. Here we mean, by organic, we mean the molecules required for life from non-organic molecules. And so what they did, Miller and Urey did, is they took gases that they believe were present in the early Earth's atmosphere, and they combined them and added electricity in kind of a closed container. When they added electricity, that mimicked lightning. And the idea behind either one of those is that lightning or electricity is a very concentrated amount of energy occurring all at once. And so that huge burst of energy could catalyze or jumpstart a number of different chemical reactions that maybe couldn't have occurred without that helping hand. And what they found is that when they added electricity to that mixture of gases, they were able to produce molecules such as amino acids and nucleotides, which was really significant considering at the time they didn't think there was any way for those molecules to be created. But it showed them, contrary to what they believed at the time, it was in fact possible to produce organic types of molecules from inorganic or non-organic molecules, or gases or elements or things of the sort. So that was a pretty important step in terms of discovering what we would consider to be chemical evolution. Now, there is an alternative hypothesis. And so to clarify, what the Miller-Urey experiment really hypothesizes is that life first started as some intense amount of energy initiated a chain of chemical reactions that created life's building blocks. And those building blocks would have combined to form life. That's the first explanation. The alternative hypothesis is the meteorite hypothesis. And so this is based on the fact that scientists have found organic molecules like amino acids on and inside asteroids and meteors that have made it from space onto the Earth's surface. They have literally been found, which suggests a few things. One, it suggests that maybe there was no chemical evolution, and actually these molecules were just brought into the Earth's atmosphere from the outside, from space, and that's how they were introduced, which, based on the meteorite hypothesis, or based on the fact that amino acids have been found on and in meteors, makes sense. But it also suggests this idea that maybe they were just present from the beginning, because if these things are already present in space, maybe when they combined into Earth, well, maybe they were just there to begin with. So either one of those kind of takeaways is based on the idea, again, that amino acids have been found on asteroids that have made it from space to Earth's surface. And so those are the kind of two leading ideas or hypotheses for how organic molecules could have first appeared on Earth. Now, the next question is, how did cells form? Because, okay, maybe I've got the stuff I need to have life. It doesn't do me any good if I don't have a way to get it together and to stay together. I think the example I used in class is, if I was going to start a bakery. Now, why would I start a bakery? A baking business. Howdy Browdy's baking. I don't know. If I had everything that I needed to form a baking business, but I didn't have a place to actually have the business, well, then I wouldn't have a baking business, right? I need a place for everything to happen. And so that's what we see here with these two hypotheses for how the first cells formed. So the first is the iron sulfide bubble hypothesis. And the idea is that kind of deep on the ocean floor, there are these hydrothermal vents that are really hot, and molten or liquid iron sulfide would come up from the ocean floor and cool. And as it cooled, it would create these chimney-like structures that we see depicted here. Now, obviously, there's a light being shown on them to take this picture. But they're these chimney-like sort of rocky metallic structures that are kind of like a tube or a chimney. The thought is that these structures might have sealed organic molecules that would have otherwise been floating in the ocean. So we believe that these chemicals, the amino acids and nucleotides and things needed for life, would have been floating around in the ocean because there wasn't really much else on early Earth. And so we believe that they might have made their way up into these sorts of chimney-like structures, and in there, those structures could have acted like a cell membrane and contained those molecules, giving them a place to interact. Because if everything's just in the open ocean, the chance of those things running into each other is relatively low. Whereas if these things are contained within a relatively confined area, well, now the chance of those molecules interacting the way that would be needed to form life, suddenly that's a lot more likely. So that's the iron sulfide bubble hypothesis. Now, the second, and maybe most familiar with what we've talked about in class, is the lipid membrane hypothesis. And so you might remember from Unit 3, cell membranes, that we talked about the phospholipid bilayer, and we basically see that here. We see two layers of lipids. Remember that lipids have polar hydrophilic, water-loving heads, and non-polar hydrophobic or water-fearing tails. So they arrange themselves because of that into two layers. The heads facing the inside and the outside of that membrane. And then in the middle, the hydrophobic tails are facing each other. So things that don't like water will be away from water, and things that do like water will be near water. Because they arrange themselves like that naturally, the thought is that lipids that might have been created in the early atmosphere, like amino acids or nucleotides, could have come into contact with one another and arranged themselves into this sort of configuration. So at some point, that lipid bilayer would have formed, and that would have separated the inside environment from the outside environment. And so the idea here is that, well, okay, this could be a way that a cell membrane-like or cell membrane structure could have formed initially. Now, obviously, there's a lot of links missing in between the start of Earth, the formation of these chemicals, formation of cells, and what we have today. And so we're certainly still discovering a lot of these things, but that's the main idea. And the main takeaway from this concept is that for life to exist, internal conditions have to be kept separate from the outside, right? You wouldn't be a person if you didn't have skin. You would just be a pile or a collection of bones and blood and organs, and that's not really that appealing, and I apologize for that. But if you think about it, really, your skin is what makes you distinct from the outside, right? If you get a cut, that is an opening from the outside to the inside of who you are. So your skin acts as your membrane. The cell membrane serves that same purpose for a cell. And so for life to occur, there needs to be some sort of clear division between outside and inside. Now, to move on in the notes, I'm going to hopefully be done with this before the 20-minute mark. Maybe even. Yeah, we'll see. So we've got the molecules needed. Okay, we've got a cell. Well, in order for life to progress over time, we need some way for life to really pass on traits. We need some sort of genetic material. And we believe that that was RNA. That RNA was the first genetic material, and we believe that for a few reasons. For one, it's simpler than DNA. It's a single strand. If you remember back in Unit 4 and 5, we talked about how RNA was single-stranded, DNA was double-stranded. DNA needs enzymes to replicate itself. We studied this for a whole unit, right? You need helicase, you need ligase, polymerase, all this stuff. Whereas certain kinds of RNA can just copy themselves without enzymes. So RNA in a form called a ribozyme is able to actually replicate itself without the help of any additional enzymes. So you can take one piece of RNA and essentially make it into two. That's huge in the early Earth. If RNA is able to replicate itself without any outside help, that would certainly suggest that it could function as genetic material early on when these complex DNA systems aren't really needed. We just need some way to pass on genetic information in the form of nucleotides, and RNA could be the way that that happened. Now, the end of the notes here kind of move all over the place, I will admit, but okay, now we've got genetic material. Well, what evidence do we have of early life? And the oldest fossils that we actually have, the oldest fossil record, or the records that we have of the oldest living things ever to be found, are photosynthetic prokaryotes known as cyanobacteria. Marine cyanobacteria just mean that they're water-dwelling or ocean-dwelling, and it's important to think about what we see. So prokaryotes, I've said already today, are less complex than eukaryotes. Prokaryotes don't have a nucleus, they don't have organelles, they're basically just DNA or some genetic material. So DNA or RNA, a cell membrane, cytoplasm, and ribosomes. Basically just the basics of what you need. Now they might have cilia or flagella, things like that, but that's a prokaryote. Photosynthetic implies that they're able to make their own food from the sun. They're autotrophs. And so photosynthetic here means that they could make their own food. Imagine being the first living thing ever to be created, or not created, to ever form, and then, okay, now you gotta eat another living thing. That'd be a cool joke, right? So it would make sense that the first living thing would need to be able to sustain life on its own. And so it makes sense that photosynthetic prokaryotes would be the first sort of life, at least that we've been able to find. And cyanobacteria actually do still exist in oceans and lakes. Here we see a picture of a cyanobacteria bloom that's caused by fertilizer, because we use fertilizers that have a lot of phosphates and nitrates, which are basically the ideal food for these sorts of cyanobacteria. And so when those run off in watersheds, you can end up with these cyanobacteria blooms, where water turns green. Now, this might be a very familiar picture if you've ever physically been in my classroom. It's been on the wall all year, but we talked about that eukaryotic cells, we believe, evolved from prokaryotic cells. That's why eukaryotic cells are more complex. And one of the ways we think that happened, or the main way we think that happened, is through what we've called endosymbiosis, or symbiosis. Endo means inside, sym means together, and bio means life. So endosymbiosis means living together inside each other. I've talked about this a few times in class already. I talked about it in the cell energy units. I talked about it in the cells unit. We think that a large, kind of early prokaryotic cell would have eaten or engulfed some form of archaea or bacteria, some of which might have been good at, not chloroplast, at photosynthesis, which would have become the chloroplast, and some which would have been good at cell respiration, which would have been the mitochondria. And we think that over time, as those engulfed, so this is what we're seeing here, the cell engulfed these and essentially became a eukaryote over time. Now, this is the very kind of simplified explanation, but it makes sense for a number of reasons. Mitochondria and chloroplasts, first and foremost, have their own DNA, which is huge. That right away suggests that those might have initially, or probably were initially, something separate. So you have different DNA than your DNA in your cells because your mitochondria all have DNA. You don't have chloroplast DNA because you're not a plant. You don't have chloroplast. Now, we also know beyond that both of those organelles also have two membranes. They have also their own form of cytoplasm, and so a lot of anatomical, kind of like structural traits of those organelles seem to suggest that they were once their own individual living thing. And with that, we will conclude this before the 20-minute mark, just about, with the fact there are three different kinds of DNA that you and plants have. Cellular DNA is the DNA that's just in the nucleus of your cells. Prokaryotes have it, eukaryotes have it. It's what's shuffled in sexual reproduction. That is just the normal cellular DNA that we talk about. Now there's also mitochondrial DNA in eukaryotes. It's passed on from the mother. And if you think about why that might be, it actually kind of makes sense. Well, what do moms and dads contribute to reproduction? Dads contribute sperm, which is essentially just a package of DNA with a little tail and like a drill on the tip to get into the egg. Now what is the egg? Well, the egg is DNA, but it's nutrients, it's organelles, it's cytoplasm, it's everything else needed for life. And so, if the egg is what's providing the mitochondria, the organelles, to offspring, you will have only your mom's mitochondria and not your dad's. And so this is actually really useful for tracing ancestry because it's a maternally passed on thing with no exceptions, which is really cool. Now, chloroplast DNA follows that same pathway for that same reason, but we of course don't have chloroplast DNA because we're not plants, we don't have chloroplast. And so neither one of these types of DNA is shuffled in reproduction. So in sexual reproduction, we see DNA being shuffled and getting randomized because we don't see that in these sorts of DNA because these are contained, right? And really, that's it for this unit. This lecture and this unit. I went a little over the 20-minute mark, and I appreciate that. I talked about a number of different things today, the history of life, different hypotheses for where we think the Earth, organic molecules, cells, things like that came from. So I hope this has been interesting. I hope this has been helpful. I hope it's been entertaining. And so, as always, again, if you have any questions, you know how to get a hold of me in person, over email, over Canvas. I'll get back to you as soon as I can. It might not always be quick, but I'll get back to you as quickly as I can. And again, I hope this has been enjoyable, and I will see you in the next one. Take care.